Knowing exactly what bases make up a particular gene allows us to make comparisons of genomes between individuals, so we can find out the causes of some diseases in personalised medicine. It allows us to trace different variants of a pathogen, so we can track the outbreak and spread of epidemics and pandemics, and it even allows us to compare genomes between completely different species, so we can identify the phylogenetic relationships and trace our own evolutionary history. Now, in order to carry out DNA sequencing, which is working out exactly what the base sequence of a section of DNA is, first we need to make lots and lots of copies of the DNA that we're trying to replicate using PCR. Once we have these template strands of what we want to sequence, the Sanger method then involves making lots of incomplete copies of that strand, and the analysis of those incomplete strands through electrophoresis will help us to work out what the bases were. Think about how normally DNA replication happens in the nucleus. You have DNA helicase, which has caused the double-stranded helix to unwind. There are lots of free complementary DNA nucleotides floating around the cytoplasm, and they line themselves up against that template strand according to complementary base pairing. So you're always going to have adenine bonding with thymine, and you'll always have cytosine bonding with guanine. And then DNA polymerase comes along to help form the phosphodiester bonds between one base and the next, to actually combine all of these nucleotides and form a new DNA strand. This process is going to continue until you've got a complete DNA molecule. And by the end of it, what you should have are two identical molecules of DNA, where each new molecule is made up of one original strand from the original DNA molecule and one completely new molecule made from the free complementary bases which were previously floating around the nucleus. So that's what we mean by semi-conservative replication. Now imagine that if, as well as the normal free bases of adenine, thymine, cytosine and guanine, there were also versions of the bases that had something a little bit different about them. And what was different about them meant that DNA polymerase could not cause these bases to make any more phosphodiester bonds after them. So if one of these strange new bases attached onto that template strand, the copy of the DNA would then be incomplete. You've made so much, but once one of these bases has attached, you can't go on any further. To carry out the Sanger sequencing method, you first take a tube full of copies of the DNA molecule that you're actually wanting to sequence. Remember, there are going to be lots of copies of this particular strand because you've made them from the PCR reaction. You'll also have lots of copies of all four normal nucleotides, adenine, thymine, cytosine, guanine. You're going to have versions of the DNA polymerase enzyme, and you're going to have copies of a primer. Now, a primer is a very short strand of DNA which is complementary to one end of your DNA fragment. That is going to attach onto the DNA strand right at the beginning, no matter what comes on after it, and that will allow the DNA polymerase to actually bind onto it to start attaching nucleotides. If there isn't a primer, then DNA polymerase is not going to be able to attach to the strand by itself. And that primer actually has a dual purpose. It also acts as a probe. If you've made it radioactive, it will help the DNA to be visible later on when we carry out gel electrophoresis. Or if you've tagged on a little bit of fluorescent dye onto this primer, then the primer will show up again after the process of electrophoresis. As well as that, the tube also is going to contain lots of copies of a special version of one of the nucleotides. In this case, I've got lots of copies of a specially adapted nucleotide version of adenine, and I'm going to call this terminator nucleotides. So these are terminator adenine nucleotides. They are modified in a way that means they can't make phosphodiester bonds. So once they've been attached, DNA polymerase can't attach any more nucleotides, and the copy will now be incomplete. Whether a normal adenine attaches to the template or a terminator adenine attaches is completely random. But because there are going to be so many copies of the template in the tube, you'll end up with a mixture of different sized fragments being produced, all of them ending in terminator A, or which will have completed the fragments. In this case, in this example, a normal adenine happened to bind on, and that means that when DNA polymerase comes along, it can continue to make phosphodiester bonds between the nucleotides, and you'll end up with a longer fragment. But now look at what happens if, when the complementary bases are lining up, a terminator adenine comes to line up with this thymine instead of a normal one. The DNA polymerase comes along, it attaches to the primer, and it forms phosphodiester bonds between the primer and the first nucleotide, between that nucleotide and the next guanine as well. It forms one between the guanine and the terminator nucleotide. But because this is a terminator, it can't make any more phosphodiester bonds at the other end. So at this point now, the DNA polymerase detaches and this strand is incomplete. It's much smaller than the first strand, which was actually finished off. Key thing to remember at this point is whether you have a shorter, incomplete copy of your DNA strand or you have a longer one, there is a radioactive primer that acts as a probe at the beginning of every single fragment. You then repeat the whole process, but this time with terminator thymine in the tube, another time with terminator cytosine, and another time with terminator guanine. In every tube, you're going to end up with fragments of DNA of different sizes, all ending in the terminator base, or having been actually completed. To simplify what this is going to end up looking like, I'm just going to show you what the replicated strand will be, rather than show it with the original molecule every single time. So if we come back to our original tube with terminator adenines in them, you will have some copies of the replicated DNA strand which are incomplete, because after the primer, which is in our example the very first 3 bases, which are always going to be there, the first place where adenine could have attached on happened to be a terminator adenine, and therefore this strand stops here. It's not going to be made any bigger than it already is. You will also have attempts at copying that original strand where you've got your primer, which is there anyway, the first adenine that could have bound on because of complementary base pairing with that first thymine. The first adenine is a normal one, but the next adenine that complementary could bind on happens to be a terminator nucleotide, so this strand is a little bit longer than my first strand, but it's still incomplete. And you will get some versions of copies of that DNA molecule where the first adenine was a normal adenine, the second one was a normal adenine, and it's the third adenine that needed to bind on after the primer, which happened to be a terminator nucleotide. Because you've got so many copies of the strand of DNA that needs to be replicated or needs to have a complementary strand attached onto it, there are going to be versions of different nuclear strands which are of different lengths, but all of them will end in the terminator nucleotide, or they might have been completed if you happen to have all normal nucleotides which are binding onto it. Once you've allowed enough time to make all of these complete and incomplete versions of the DNA molecule, you then take all of the fragments that you had from the terminator adenine tube and you place them into the first well of an electrophoresis gel. When you run an electrophoresis gel, all of the DNA strands will separate according to their size. So the larger fragments won't travel as far. The shorter fragments will be able to travel a little bit further. And you end up with a separation pattern on your gel electrophoresis according to the size of each of these fragments. So you can see which one is bigger. In one of the other wells in the same gel electrophoresis, you will also have put in a DNA ladder, which is a mixture of fragments that you know how long those fragments are. So you will have put in a fragment which you know is 15 bases long. You will have put in a fragment that you know is 10 bases long, and a fragment that is maybe 5 bases long. And it's not going to be these kind of numbers, but this just gives you an idea of what's happening. Now, if this strand here has travelled the same distance as a fragment in your DNA ladder, which is 15 bases long, then you know that this strand is 15 bases long and it must end in a terminator adenine, which means that 15 bases along your original strand, there must have been thymine. If you have a strand here, which has travelled the same distance as your DNA fragment from the ladder, which is 10 bases long, then you know that this second strand that I've just arrowed must be 10 bases long, and because it's in the terminator adenine tube, it must have ended in terminator A. Therefore, the original base at 10 bases along your original strand must have been thymine. So you can use this to work out what the original DNA sequence must have been based on how far along the gel electrophoresis each fragment travelled, because if it's in the terminator adenine tube, then it must end in terminator adenine. Therefore, the original base was thymine. If you then run the same process through for the fragments you obtained from the tube containing terminator cytosine, then all of the different fragments must end in terminator cytosine. Therefore, you match it against the DNA ladder to see how far along the bases are. If it ends in terminator cytosine, then at that position on the original strand, you must have had terminator guanine. And you repeat the process for all 4 of your terminator bases. Here's what a mini version of that electrophoresis gel might actually end up looking like. So in this well, you put in all the fragments that you got from the terminator adenine tube. That means that every single one of these fragments must end in terminator A. In the second well, you've got all of the fragments that were obtained from the terminator thymine tube. So everything in this well must end in terminator thymine, and so on. We know that the shortest fragments will have travelled the furthest distance, and the longest fragments will have travelled the shortest distance. So now we just need to read where those bases have appeared in order to come up with the original sequence. The shortest fragment ends in thymine, which means the first base that I'm reading off here, I've got terminator thymine. The second fragment, in terms of the length of the fragment, ends in terminator adenine. Then you've got thymine again. And I'm just reading the bases travelling up the gel. Excuse the handwriting. Now, if these are the terminators at the end of the strands, that tells me that the original strand must have had a base which was complementary to these ones. So if the shortest fragment on this gel ends in thymine, then the first base, not counting the primer, the first base on the original strand must have been adenine. And the second one must have been thymine, the third one must have been adenine, and so on, so that I am just reading the complementary bases. This is the original base sequence. Remember that the only reason that these strands on the gel electrophoresis are showing up is because every single fragment had a primer attached to the beginning of it that had been labelled either radioactively or fluorescently. So when you look at the gel afterwards, what you can actually see are everywhere where you have a fragment that starts with a probe, a labelled primer, that shows up when you place a photographic film over the gel after it ends. The original Sanger sequencing method would have to be done for each bit of DNA that you wanted to sequence in 4 different tubes with the separated adenine, thymine, cytosine and guanine, because originally all of those probes were radioactively labelled. So if you didn't have terminator adenines in a separate well to terminator thymines, you wouldn't be able to tell which one was which. The whole process was made much faster and simpler when scientists stopped labelling the probes with radioactive molecules and instead tagged them fluorescently instead, because now every single terminator nucleotide is tagged with a different colour of a fluorescent label, and you just shine a laser through to work out what colour each one is, so they no longer need to be separated into different tubes. This meant that the original Human Genome Project, because of these advances in the techniques that were being used, ended up being completed faster than the original deadline and under budget, which hardly ever happens for any kind of large-scale project. For the OCR specification, you need to know how Sanger sequencing method was used to sequence DNA, how improvements in technology made the whole process much faster, and be able to answer questions related to the process, which includes being able to read the electrophoresis gels. I've put the link to the video, some slides to go alongside it, and some practice questions for you guys to do, to have a go at inside your team's online notebooks. I'll also ask Mr. Patcher to put this video onto the school's YouTube channel. Please come to the first lesson after the holidays prepared with any questions that you have.